Neosaxitoxin is this molecule here. It's actually a natural product from microalgae, and it's currently under clinical development as a long-lasting local anesthetic. Current local anesthetics are like bupivacaine, are structurally similar to cocaine. And these molecules block voltage-gated sodium channels and thereby inhibit the transduction of nerve or action potentials. But in order to exert the effect, they have to cross the cell membrane and then lodge themselves inside the central pore of the sodium channel when it is in an open state. There's a whole range of local anesthetics which are pretty similar in structure and function. And but what is common to them is that their action lasts for maybe 2 to 6 hours, and after that you can't use these local anesthetics over prolonged periods of times because they've got quite a high intrinsic toxicity. Neosaxitoxin, on the other hand, is a very polar molecule which binds to the extracellular side of the sodium channel. And it also inactivates the channel and blocks pain, but it is a much more potent channel blocker than bupivacaine. It's about 1,000-fold more active. So the dosages you require is in the microgram per kilogram range as opposed to milligrams per kilogram for existing local anesthetics. But what is most important is that neosaxitoxin, that a single dose can block pain for a matter of days, up to 4 days. And that has quite important implications for pain management, such as in surgery, where local anesthetics are used during the surgical procedure, and they block pain immediately after the procedure, but then if post-operative pain persists, and which it often does for about 10 days, then the only class of drugs that are available to treat the pain if it's strong are opioids. But opioids have severe side effects, and they're not always efficient. And these side effects can lead to addiction or cause many accidental deaths every year. So there's a very strong incentive to replace opioids with alternative drugs. And neosaxitoxin might be such a drug. It can replace both current local anesthetics, and because of the long-lasting action, it eliminates the need for opioids. The clinical trials that have already been performed in humans indicate that it is a very safe drug. It has no serious side effects. It reduces pain and speeds up recovery of the patients. But there's one serious problem, and that is the supply. At present, there's no large-scale process for the production of neosaxitoxin. The natural producer organisms are microalgae, and the ones that do produce these compounds, they are quite fragile and they're not suitable for large-scale cultivation. And the molecule itself is quite a complex molecule. It's got 3 chiral centers. It is highly oxidized, very polar and nitrogen-rich, and it's simply not suitable for a large-scale chemical synthesis. You require over 20 steps, and you have a very low efficiency. So we are developing a solution to this shortcoming, and that is by expressing the biosynthesis genes for neosaxitoxin in E. coli. And E. coli is a common organism, of course, that can be cultivated by large-scale fermentation. We have, in our previous work, uncovered and characterized the biosynthesis gene cluster for neosaxitoxin. Initially, this was done in a cyanobacteria called Cylindrospermopsis raciborskii. And this gene cluster is about 35,000 base pairs in length, and it encodes many of the enzyme families that we predicted would be present in this pathway, but in addition, a lot more. And there are still a number of genes in this pathway or in this cluster where we don't know what their function is. This is counting down, right? Okay. However, we have identified the core biosynthesis genes and confirmed their function by detecting the intermediate metabolites, and some of them were also expressed in vitro and biochemically characterized. So we have a good idea about the pathway. In order to characterize the gene cluster and also to produce Neosaxitoxin, we tried to clone the entire gene cluster into E. coli. And even after replacing the cyanobacteria promoters with ones that work in E. coli, we could not achieve any Neosaxitoxin production. And that is the reason why we resorted to gene synthesis. So instead of using the native genes, we designed expression constructs that were optimized for expression in E. coli. And some of the optimizations we did was we replaced the promoters. We chose the PM promoter, which Trygve knows very well. We fitted every gene with a synthetic ribosomal binding site, which we thought would be a strong ribosomal binding site and drive translation. And we used the algorithm provided by the gene synthesis manufacturer to codon optimize the genes. Because this was in 2014, it was thought that low-frequency codons could interfere with efficient translation of proteins. We integrated the constructs into E. coli, into the genome, and then optimized the culturing conditions, which are pretty standard, and we could detect Neosaxitoxin production. We used LCMS, an ELISA method, and a functional assay, and all three assays were positive. But the levels of production were very low. So currently we are working on optimizing Neosaxitoxin production on several levels. One is that we found that precursors were limiting, such as acetyl-CoA and carbamyl phosphate. And one thing that is very interesting is that the genetic elements that we used did not perform as we predicted. The proteins in these multi-gene constructs are not expressed, not nearly expressed at the same ratios as in the native organism, and that may lead to metabolic inefficiencies. And also part of the proportion of the expressed proteins are not catalytically active, which may indicate that the codon optimization is not optimal anyway. So we're looking for a biophysicist to help us with these aspects. Once we have a good producer strain, then we can move forward to optimize culturing conditions and develop a full-scale bioprocess, which of course will take time. And there are many people involved in this research project, which I would like to acknowledge. And we also have funding from the Norwegian Research Council with a biotech and a FORNY grant. Yes, and I'm out of time.
Thank you very much for this very interesting talk. So any questions from the audience? I can start then while they are thinking. So 12 genes, 12 enzymes or more you have to express in E. coli?
It's about 20 because we have included the ones that we know are required and ones that may be required.
Mm. But I mean, as long as you detect your product, which definitely shouldn't be there naturally, you have a proof that somehow everything works. In principle, it's the low level could be due to only one enzyme produced at very low levels or not very active. So how do you attack to find the bottleneck in the biosynthetic pathway that you have constructed in E. coli?
We've got a pretty good idea where the bottlenecks are, and they are in the expression of the genes and in the precursor supply. But also, we've got a challenge that we have to match the production levels of the algae, and we are magnitudes of that. So it's not just one enzyme or one gene which is limiting production.
You know that there are more.
Yeah.
What about the intermediates? Could they somehow interfere with the metabolism in a not so good way for E. coli, and that could give you problems?
We don't have any indication that they could interfere. Most of the intermediates are actually depleted. There are a couple of intermediates which accumulate in the pathway. The growth rate of the modified strain is similar to the native strain, so it doesn't seem that it's repressing growth in any way.
And the product is not toxic?
No, they are, E. coli is a prokaryote, and Neosaxitoxin is a eukaryotic voltage-gated sodium channel blocker. No, very specific.
Anyone else? Otherwise, I say thank you very much once more, and a present also for you.
Thank you.
Thank you.